welcome today in our BioNeex expert interview Dr. Hitesh Mistry from Physiomics. Dr. Mistry will explain what is the best way for developmental stage biopharma companies to present their data in oncology. My name is Marie Vosny and I have a background in molecular biology, especially in oncology, and I would like to start with a short introduction of our company BioNeex and then Physiomics. BioNeex is an online peer-to-peer platform for R&D stage licensing and collaborative partnership in biopharmaceutical industry. The BioNeex platform opens the unique opportunity for developmental stage biopharma companies to display their novel compounds, for example, on a global scale, and on the other hand, allows biopharma companies to expand their R&D pipeline by providing direct access to innovative compounds available for out-licensing and co-developmental partnerships. Our partner, Physiomics, is a solution provider to R&D-based pharmaceutical companies with a focus on oncology, and the company uses a combination of industry standard and its own virtual tumor modeling technologies to predict the effects of cancer drugs and treatments to improve the success rate of drug discovery and development while reducing time and cost. Our guest today, Dr. Hitesh Mistry, thank you for joining the interview, is the principal consultant of Physiomics focusing on clinical modeling. He's a specialist in simulation and has a background in mathematics. Additionally, he is a senior research fellow at the Manchester University, and he's on the editorial board of the International Journal of Radiation Oncology. Dear Dr. Mistry, my first question to you would be, how does Physiomics support small companies developing cancer drugs?
Thank you. So I guess our core expertise has been in cancer drug development for a number of years. So since the company was first incepted about just over 15 years ago, the focus has always been on oncology. So this kind of tells you a bit about the sort of people that work there. So the whole team and all its consultants generally have a huge extensive experience of oncology drug development. And this ranges from right at the beginning, from drug discovery through to preclinical development and all the way through to clinical development and now even into clinical practice. And so the main purpose of Physiomics was to always provide what we call mathematical modeling and simulation support. So originally the company started off originally in the field of systems biology. So usually to help companies to sort of understand their drug targets and being able to sort of suggest biomarkers that could well be useful for future development. More recently, I think their focus over the last 10 years has been more towards what you'd call sort of in the preclinical sort of PK/PD or pharmacokinetic pharmacodynamic space, as well as the clinical sort of space for the same sort of modeling techniques. And this is a mix of sort of mathematical and statistical techniques that they use. So they use standard sort of tools, so the standard sort of PK/PD modeling software that does exist and models that you can use. But they also create their own sort of bespoke software, which is called the virtual tumor. And I'll tell you a little bit more about that later. But in addition to those things, we've started to branch out a little bit more into, I guess, the fashionable things like machine learning and artificial intelligence.
Yeah.
So everyone's taking a look at that, and so are Physiomics to see what's interesting there. Can it be useful? Is it better than what we currently do? So we're interested in benchmarking these methods too. So back to the main product of the company. So how do we support sort of small biotech companies? So with virtual tumor mainly, the idea is to be able to sort of explore combination schedules. So the idea is to sort of say, well, you can't really explore all the possible combinations you want to explore, you'd like to do, either in xenograft studies or in clinical trials. So you're usually given some prior data, some idea of the monotherapy benefits, and maybe you've done one combination study. So the idea of virtual tumor is to sort of take that information and then to sort of say, well, what is the optimal schedule? And so the optimal schedule here is kind of a schedule that balances both sort of safety and efficacy. And by hopefully coming up with the optimal schedule quicker, a company will be saving time, which is quite useful in this day and age where everyone's going to rush to market to the newest combination.
Really important, yeah.
So the idea is to save time, and clearly there's a bonus to this. You save animals and you save costs in terms of experiments.
Yeah.
I guess although a lot of the virtual tumor itself started off in the preclinical space, so the whole idea was to always work with xenograft data. mainly because of throughput originally. But over the last 4 or 5 years, we've moved more and more towards the clinical space. And this is where we support companies in the early phase sort of drug development to sort of make decisions about what dose to use in combinations, and then to start thinking about combination schedules. So again, on top of that, we kind of offer more of a, I guess, a general sort of data analysis approach too for biotech customers. So I guess we have two sets of clients, sort of large pharma companies who are more interested in the virtual tumor technology. And then we have, I guess, more smaller biotech companies who don't have any sort of data analysis support internally.
Mm-hmm.
And so for those companies, we kind of not just apply the virtual tumor technique, but also help them purely with data analysis. So help them to understand what data they've got. Are there any missing gaps? So do all the data sets actually make sense? If they don't, why don't they make sense? Can we generate hypotheses? Can we generate experiments that would help them? And the idea is to essentially pull together all their data and give them a story. And the idea is that this story will help them with gaining funding for the next stage of development or find possible partners.
Awesome. That sounds really promising. Thank you for explaining that. Yeah, my next question would be, what do you see as a good set of preclinical data, or do you have any tips what people should avoid by presenting their data?
It's quite a good question. So a good set of preclinical data, I guess, there's a lot to consider, right?
Yeah.
So I guess from an efficacy point of view, I guess you want to see a causal relationship, right? So maybe you have an idea for a target. So it's a good idea to show that if you have developed a molecule for a target, that your molecule does actually engage with that target, right? So a nice example of that could be the MAP kinase signaling cascade, which people know quite well. So say if you have a MET kinase inhibitor, I think people know the downstream endpoint is ERK kinase. So a nice thing to be able to show is if you have a MEK inhibitor is that you actually affected the phosphorylation of ERK, which is due to MEK, right? And hopefully show it in a dose-dependent manner. So increasing concentrations, you see an increase in sort of effect. So being able to show that kind of consistent data. And then on top of that, you kind of want to know, well, okay, that's all well and good. You've shown that your drug actually engages with the target, but then can you also elicit the phenotypic effect you're interested in? Right? So can you show how your drug actually affects cancer cells from growing? It may well be cell cycle arrest, apoptosis, or other effects. But being able to pull together this causal chain is quite important, I think, to be able to sort of say, yes, my molecule is doing what I think it's saying, what I said it was saying. The molecule does sort of encapsulate the ideas we had about it. And I think that's quite important. And usually you can do this just purely with in vitro data, right? So there's some simple experiments you could do. And then usually the next stage I find is clearly we'll move on to xenografts. So the in vivo setting is clearly quite important. And quite often what we used to see when we used to go to conferences, used to talk to people, they would show xenograft experiments, and sometimes they would show their compound and say, well, our compound's not working, we're not seeing an anti-tumor effect. And the first likely question to ask is, well, actually, is the drug there? So when you've administered the drug to these mice, do they have exposure? Have you measured it? And you'd be surprised how often people haven't actually thought about some of the basics, saying, well, I've dosed to a mouse, and they just kind of assume that the drug is there and it's there on target, but they've never actually measured plasma concentration to sort of assess whether there is any exposure. So that's one of the first sort of in vivo things to check, right? And larger companies would do this routinely. I guess biotechs don't always think this way if they're on a quick way to trying to quickly progress a compound. And then there's some more typical questions you could ask about the pharmacokinetics. So, does the exposure change with increasing dose? You hope so. You hope you see as you increase dose, you increase exposure. On repeat dosing, so at the end of your xenograft study, say you made it a 14-day xenograft study, you're dosing every day. At the end of the study, do you still see exposure? So if you were to take mice and sort of assess sort of drug levels, do you still see it? So this kind of gives you an idea of whether or not the drug is being cleared through certain different mechanisms.
Yeah, okay, I totally agree. I think your points are very important, especially to set up your experimental data set, or to set up just the experiment and make sure that everything is in the right place, and then to start working on that. But first, make sure that everything is just in the right cases. So very good points. Yeah, this is very important to us to present the data in a good way and to have this good setting, because one of the value proposition of BioNeex compared to other databases, for example, is that our compounds provider who provide their compounds have the exclusive privilege to list their compounds on the platform themselves, and they also have the opportunity to upload a non-confidential information or package set about their compounds or their company. And yeah, it's very important that they know how to present their compounds in the right way, because this is fundamental in order to find a licensing partner or a collaborative partner for themselves. Awesome, good. My next point would be, is there anything that if you see it immediately raises issues in your mind when there is a data set and there are special points, yeah, you are just familiar that it might be some problems or there might be some issues with it? Is there anything like this?
Yeah, so I guess we always look for data consistency. So if people have done quite a few experiments, and sometimes the repeated experiments, we'd start to look at variability. And sometimes what you find is that sometimes if data looks extremely noisy, far noisier than what you're used to, for example, xenograph data, people have typical xenographs that they use. And if you compare the control curves they're seeing to literature data, if something looks erratic, that kind of triggers a question about the quality of the data that's been generated. And similar for exposure response in like in vitro assays, we have seen situations where people have tried to generate exposure response, and what you tend to see is something that doesn't look like an exposure response. So you get really erratic behavior from one concentration to the next. So this kind of starts to sort of trigger warning signs in myself to sort of say, well, what's going on here? Is there some issue with the compound in terms of precipitation, or is there some issue about solubility with that compound? So it starts to trigger questions, I think, about the data. So consistency is important, and also how you present it, right? So there are a lot of tricks you can use to make your data look good in terms of sort of transforming the data in certain ways, which we've seen a lot of at conferences. So one thing is to try and avoid those tricks, right? So, for example, when you're plotting data, to always include the origin. That's usually a standard thing you should do, rather than starting the axis at some point that's interesting to you, or transforming the data in some way that accentuates your efficacy. Those are usually warning signs that you're trying to make more of the data than what's actually there.
Yeah, okay. Thank you. And my last question is, how does a company set itself up to have the best chance of being able to successfully translate its preclinical program into the clinic?
Okay, that's a very good question. I think being able to make clinical predictions from preclinical data is the hardest thing you can do in oncology. I guess for any small company or any company, I guess the most important thing is, do you understand your compound? So have you been able to show that, like I said before about target engagement, do you also understand your compound from a genetic background? So when you screen your compound against panels of cell lines, do you understand what genetic factors are really important for that drug to work in a setting? Do you understand the prevalence of that genetic factor in the clinical setting? So if you believe your drug, if you believe the genetic marker that's important for your drug is really prevalent in a certain disease setting, so maybe second line, I don't know, lung cancer, it may well be a good idea to target your phase 1 program in that setting to help you give you the best chance of starting to collect data that's going to be important for you to make decisions. So I think proactively thinking about your line of sight, right? So where's this compound going? What do I know about it already? Those things are really, really important, I think. And then I guess it then comes back a little bit to sort of say, well, you've clearly done preclinical experiments that kind of are meant to mimic your clinical experiments. So you may well think about, well, I've started to choose xenographs. Were these the right xenographs? Are they actually related to some degree to the clinical situation? And clearly you can't do a great job, right? So there is no magic bullet in terms of the right xenograph model, but you can start to think at least, well, does it have the right genetic background for what I'm interested in? So just generally just thinking, I guess, what's the word? I guess the word is in a proactive way. So thinking about the line of sight it's just really important.
Okay, good. Thank you very much, Dr. Mistry, for educating us and how to present our data in the best way, and which is very important for us and of course our clients to make their preclinical and clinical stage discoveries an attractive licensing target from pharmaceutical companies' perspective. It was a great pleasure talking to you today. And if our viewers have any questions regarding data presentation or interest in Physiomics or BioNex, you are very welcome to contact us. Thank you very much for joining and listening, and all best, and see you in our next upcoming expert interview.
Thank you.